So yeah, fortunately that we published a paper on the Nature method and also the DeepMainmast. So this new modeling tool also will be published in the Nature method. So if you are interested in the details, so please access to the Biophysics. So last week we posted the preprint on the Biophysics. So maybe you can start the DeepMainmast, Genki Terashi, then you can find the Biophysics paper. Okay. So yeah, this is a brief introduction. So yeah, maybe we already know that the number of the available cryo-EM data is rapidly increasing. And also a very interesting thing is that the resolution point of view, so a lot of high resolution cryo-EM maps are available. So that means we can use the atomic model, or we can use the atomic model built from the cryo-EM map. But we also know that in the PDB entry, some entries, atomic model have error. Yeah, very critical error. So sequence shifting or amino acid assignment is not accurate, or position is incorrect. So in that case, we developed the DAQ score to evaluate how the accuracy of the atomic model. And also we built a DeepMainmast, which is a deep learning-based model, de novo modeling method. So from the cryo-EM map, we can build atomic model. And also we recently published a paper, the cryo-EM. So last week, the shower presented. So from the EM map, so we can build the RNA, DNA structure. So today I will show the DAQ score and DeepMainmast. So what is a DAQ score? DAQ score is a quality assessment method using deep learning. So suppose you have an atomic model from PDB or some other program, then you have a density map. Then using the deep learning architecture, so each 11 by 11 by voxel, so deep learning can predict the local structural features, such as secondary structure or atom type or amino acid type. Then it can compute the probability values of the atom type, amino acid type, and helix, beta coil, secondary structure information. Then we can compare with the deep learning-based prediction and actual atomic model. If this atomic model have a high agreement between this predicted local feature, so that means that this model is very accurate. But if it does not agree, so that means highly possible that this atomic model is not accurate. So for example, this is the actual example taken from the PDB database. So after using the DAQ scoring function, DAQ score says that this red region are highly possible long sequence assignment or position connector. The blue region is a high quality, DAQ score is high. So that means this atomic model at that position is rare. So we last year we published a paper on the Nature method, so it is very useful to evaluate the accuracy of the model. So why did we develop this program? So because we noticed that some PDB entries have a significant modeling error. So this is a very interesting example. Yeah, maybe you can start on the PDB website. So 6L54 and 6D and 3L3K. So these two models have a completely identical sequence, same protein, same sequence. Then it looks fitted very well to the density map. But interesting thing is that these two models have an inconsistent sequence assignment. So that means the 425 is modeled here in the blue model 6L54 and 443 the model here. That looks nice. But also interestingly that this magenta structure these three are off. So number 4 to 5 is a model here. Then 4 to 3 is model here. So it's huge sequence difference. But both structures, both PDB entries are deposited in the PDB. We don't know which model is the reliable one. Well, both models are not reliable. So then, if we use the DAQ score, it shows a significant difference at this position. So DAQ score says red color here, so that means that these positions are highly possible long sequence assignment. So this is actual example, another example. So in the PDB database, so sometimes the PDB entries have a different value. So this is a EMD 22458 resolution around 3 angstrom. So it's relatively high resolution EMR. Then the author deposited the atomic model in the 2020, October 28th. So it's recorded as a version 1.1. But later, 2021, the author updated the PDB entry, so PDB coded. So overall, the structure looks almost identical. So in my eyes, I couldn't recognize which part are different. So but if we carefully check the coordinate and sequence assignment, so these magenta regions are updated. So that means the sequence are shifted to the N-terminal region or C-terminal region. So this thing happens. But this data is very useful. So because the author updated this coordinate to the here, so that means that these regions are improved by the author. So to confirm the performance of the DAQ scoring function, so we use this kind of the two version PDB engine. Then compare with the previous for initial deposition and device model. Then how the DAQ score looks like. So actually, after computing the DAQ score says that these positions are low in the version 1. Then version 2, the DAQ score says that almost all regions are correct. So red region is a low quality region, then blue region is a high quality region. So if we check these DAQ score result and inconsistent position, that means updated position, it's very clearly correlated each other. So because the author updated this region, so that means that the version 1, the quality is not. Then it's agreeing with the DAQ score. DAQ score also says this position is not accurate. So there are several interesting examples. So left column showing that between the two versions of the atomic model PDB entries, where are updated by the author. Then this is the result of the DAQ score on the first version model, and like column showing that the device version. So clearly, we can see that the device version are higher, better DAQ score at the local position. And also they're clearly correlated with the updated position. So if the DAQ score says low score, at the same position, the coordinates are updated from the initial to the reverse version. So we plotted all positions where the inconsistent positions and consistent positions at the first version. So consistent positions with these two version models, coordinate or sequence assignments are not updated. So that means the author didn't have to update the position. So we can rely on it. We can trust that one. Then red region is the inconsistent. So like this magenta region, it's updated position. Then compute the taking the DAQ score on all positions. So yeah, clearly we can see that the inconsistent positions have a lower DAQ score. Then consistent positions have a higher DAQ score. And also this plot is showing that if the DAQ score is lower than negative 1.0, like this, that means that it's highly possible that that position is inactive. And also if the DAQ score is better than 1.0, so that means that this position is highly reliable. The good thing is that this DAQ score is already normalized. So that means that even in the different local resolutions or different quality, but always we can use these absolute values. So basically, if the DAQ score is lower than 0, negative value is bad, then positive value is good. It's very easy to understand. Positive is good, negative is bad. So this program is available online, so Google Cloud version. So later, I will show how to use this Google Cloud version. So user can easily upload the map and PDB structure, then click the button, then it will show the 3D structure, then where is the high quality region, where is the quality, and also plot the 2D plot. And also, we also released the server version. So if you want to compute the huge density map and huge atomic model, so Google Cloud has some limitations, 4 hours computation limitations. So, but in the server, there's no limitation. So you can upload the file, and also file transfer is much faster than Google Cloud. So if you have a bigger map, please try to use that. I will demonstrate this server. So DAQ score paper was released last year. Then some experimental groups already used to evaluate their model. So this is the one of the paper itself. So they solved this new EM map, then to confirm that the accuracy or reliability of the atomic model, they plot the local cross correlation values and also DAQ score. So in this case, the local correlation is better than 1.0. So that means that,
we're going to use to determine the structures.
Okay, another point. So, what actually affected is strength on the resolution band 2.5 to 4.5. Then we confirmed that up to 4.5 it's but 5 angstrom resolution, 6 angstrom, 7 angstrom resolution, it's hard. The different confuse that quality. So for example, in this case, here, so these regions doesn't show the clear color. So maybe around 0. Here around 0 value. Then also, if we check the density map carefully, so these regions are not clear. So maybe local resolution is lower than 5 or 6 angstrom. So in that case, DAQ score says 0. 0 means that I don't know. I cannot distinguish this is a good or bad. But this is reasonable. So because the density map itself does not have information of that residue type or atom information. So, so if you have any questions, please raise your hand. Then, I will put answer. It's okay so far. Okay. Then also this is another paper. So Eli paper. So they show the 2 models. So light panel showing that the previous PDB entries, they show the many red regions. So in the paper, they claim that this previous data, this PDB entry is not reliable. So DAQ score is around 0. Around 0 means almost random. Yeah. But their model, their new model, for much better DAQ score. So DAQ score was used for the structure validation. So it's already used in some paper. Okay, so yeah, DAQ score can tell us where the bad region and where the good region. So then we also published other papers, the DAQ refined. So based on the DAQ score, so because we can know that this position is not reliable. But the next question is that how can we improve that bad region? So then we developed the DAQ refined. So yeah, we are proposing that first checking the accuracy of the DAQ score of that local region, then use the DAQ refine. For DAQ refine, currently we are developing the server. It's fully automated. It's including the alpha code, prop code, and sequence alignment and DAQ score. Then it will define the structure. Then define the structure, then check the DAQ score again. So this DAQ score can be used for the modeling refinement. Yeah, and also you have some other methods, so CRUD or Phoenix. So once you build a structure, take the models by the DAQ scores, then define some other method, then check the DAQ score. Then you can improve the model accuracy, iterate. So today I don't show the demonstrate the DAQ refined server, so because I don't have enough time. But this I will show the basic idea. So you have a density map and atomic model. Then DAQ score tells us that where the good region, where the bad region. Then once we know the bad region, we DAQ define automatically remove that bad region. Then use as a template. So good region we keep the good region, but bad region we remove that position. Then use the customized alpha code. So because alpha code can be used with customized template and also customized MSA, multiple sequence array. Then we can build the different structures. Then evaluate by the DAQ score. Then we can look out for it. So this paper is already published. So this is an example. So initially, the initial models have many bad regions, but by applying the DAQ refine, it automatically refines the regions, then all looks good. Okay. The next topic, the DAQ score database. So this database is also released this year. So paper is published on the meta method recently. So this database provides the pre-computed DAQ score. So if you know that some PDB ID or EMDB ID or some keyword, you don't have to compute the DAQ score by yourself. So because the DAQ database already computed for customers. So using the keyword search, you can easily find any PDB entries. Then also it's for the local quality of the DAQ score graph. Then you can use some other things like English relations or ligand docking and so on. Okay, so this is a taken from the DAQ score database data. So because we are computing the DAQ score for almost all PDB entries, so we can find a trend of how many PDB entries have a significant modeling error. So it's very interesting. So if we use a 0.5 cutoff values, so roughly speaking, the 6% of the PDB entries have a significant modeling error. So it's kind of shocking results, but it's real. So that means that even the atomic models are deposited in the PDB database, but we need to be careful to use that atomic model. So 6% in the PDB entries are something wrong. Okay, so the next topic is the DeepMainmast. So DeepMainmast is a protein structure modeling. So actually, this method is based on the DAQ score. So by using the deep learning architecture, we can estimate that this position should be the what kind of amino acid or atom type. So based on that information, we can not only evaluate the atomic model, but also we can build the structure. So idea is almost same with the Cryo-EM. So first, we have a density map. From the density map, we can estimate the amino acid types for each position and also the atom type. So green dot showing that the C alpha position. Then from that information, we use the computational optimization method to DAQ routing problem solver, try to find the path. So from these dots, so computer program try to connect all of. Then we can get a lot of fragments, lot of the paths with the equal. They combine all structures, combine them, build the final model. Always we check the accuracy for DAQ score of the local positions, then try to build the entire regions based on the DAQ score. Okay, so the paper is now in place in the meta method, but the preprint is available in the bioarchive. So if you are interested in it, please check our preprint. So one of the interesting new features is that DeepMainmast also can consider the AlphaFold2 model. So because now people use the AlphaFold2 model, it's very accurate, but sometimes the local conformation is different. So like two domain structures, AlphaFold generates a packed structure, but the density map showing the open structure. So in that case, so this DeepMainmast automatically identify that the corresponding positions, then give the open form from the code form. So this is a new feature. So we tested the performance of the DeepMainmast. So DeepMainmast base is without using the AlphaFold2 model. Then we compare with the same benchmarking test. So CRI taser, this is a template-based method, search against a database, then use the atomic models, then fit the density. So CRI taser is currently one of the best template-based modeling method. Then of course AlphaFold2. So yeah, in the DeepMainmast base, the number of the better modeling higher is better. So our DeepMainmast base achieved a better result than the CRI taser, and also that it's almost same, but slightly better result than the AlphaFold2. But yeah, we can see that a lot of targets, difficult targets, AlphaFold2 achieved better results than the DeepMainmast. But if we use or if we use the AlphaFold2 model, then compute the DeepMainmast, then performance was dramatically improved. Yeah, almost, yeah, all targets of DeepMainmast achieved better than the CRI taser and also better than the AlphaFold2. So that means that even the global structures are different, but AlphaFold2 models can help us to build the accurate structure. Okay, so yeah, this is a typical interesting example. The magenta structure is a PDB models, deposit models. Then rainbow colored model is a predicted model by the DeepMainmast base or DeepMainmast with AlphaFold2 model. So this is AlphaFold2 model. So AlphaFold2 models have a closed form, so domains are closed. But the DeepMainmast can build open form structure. So because DeepMainmast taking the correspondence between the each position with the density map. Then that can give a new structure. This is another example. So it's a relatively difficult structure. So it has a long beta speed structure. Then AlphaFold 2 gives the very different structures. But DeepMainmast can give the accurate structure because it's based on the density map and DAQ score, always checking the DAQ scores to achieve the better DAQ score. And also the DeepMainmast can handle the multiple sequence alignment. So in this case, 1, 2, 3, 4, 5 chains. This is a native structure, deposit the structure in the PDB. Then DeepMainmast assign the chain ID also. So I compare with the DeepRacer. DeepRacer is also the very nice modeling tool, but sometimes it's a very fragmented on chain assignment is not accurate. So in that case, we achieve the better chain assignment and also the team score, better modeling results. Then related with the previous questions, so local resolutions. If the resolution is low, how it looks like? So DeepMainmast based and DeepMainmast. So in the both methods, so if we compare with the local resolution, x-axis and local resolution, and y-axis the accuracy of the modeling CFR position assignment. So if the resolution is better than 3 angstrom, so almost 98% of the positions are correctly modeled. But 5 or 6 or worse than the 6 on resolution at the local, then performance dramatically drop. So this is also same as the amino acid type assignment. If resolution is better than 3 angstroms, so deep learning can assign the amino acid very accurately, but 5, 6 worse than 6 angstrom, the performance the dramatically drop by the blue bar DeepMainmast test. But orange bar showing that the DeepMainmast with AlphaFold. So AlphaFold 2 help us to assign the more accurate amino acid type. It's some drop. We can see some drop, performance drop, but it's better than the DeepMainmast test. So this is one of the typical examples. DeepMainmast based and DeepMainmast with AlphaFold, and this is the AlphaFold 2 model. So resolution is a 4.1, but the local resolution is a much worse, maybe 4.5 or 5 angstrom resolution. So DeepMainmast based generates some strange connection. The RMSD is 12.7. If we check the AlphaFold 2 model, so overall structure looks correct, but the relative position of the helices are different. So some helices, for example, this blue and cyan helices are shifted to the up. Then this red helix is go outside. So overall structure looks correct, but detail is not accurate. Then DeepMainmast with AlphaFold 2, it's kind of the combination of the DeepMainmast base and AlphaFold 2. They fix the position of the helix position, and also the sequence assignment is much better than the AlphaFold 2. So this is one of the advantages of using the AlphaFold 2 model. Okay, it's 30 minutes. So summaries, yes. So we developed a new tool for DAQ score and DeepMainmast for using the deep learning method. So DAQ score is a very powerful tool to evaluate the local quality of the model. And also we also released a DAQ score database. So if you are interested in some specific PDB ID or EMDB ID or some keyword like SARS-CoV-19, spike protein, like so on, you can take the accuracy in the structure. So currently, 172,000 models. So, but last week we updated the entries, so maybe we have more entries. So, what you can search the structure. And also, yeah, I briefly demonstrate, show that the algorithm of the DeepMainmast. So if you use the deep learning method, path tracing, and also can consider the AlphaFold2 models. And this currently the performs better than the existing method. So all data are published in the Nature method and bioarchives. So yeah, data, yeah, please access to the website. Okay, so then now we can demonstrate the software. So all programs are available in this Kihara EM suite or server. So it's EM Kihara. So if you have a lab, yes.
Are you already recording?
Yes. Okay. Okay, so now the server is also available, so it's free server. So you can register your name, then you can manage your jobs. So yeah, we have lots of programs available in this web server. So Cryo-EM is available. So last week, yeah, Shao showed that the DeepMainmast, DAQ scores, and also DAQ define is available. Okay, so yeah. I want to say thank you for the this project, Professor Kihara. Then also the Shao, he developed the deep learning architectures and also server and Google Cloud part. Devashi also developed Google Cloud version. And he developed the DAQ score database. And also the thank you for the startup members. Okay, let's do the demos. So DAQ score, Google Cloud versions. Yeah, if you have a laptop, yeah, please access to the here. And also I will show my screens, how to use these ones. So yeah, these are brief bullet points what we want to show today. So first, I will show the DAQ score Google Cloud versions. Also, I will show some details in the screen. So first, I will show the screen slide. Then next, I will show the actual demonstrations. So DAQ score Google Cloud versions. So yeah, last week I found that a very interesting entry. So EMD 31563. This is the spike protein of the COVID-19 virus. Then PDB entry is a 7 FETs. So let's check these models. Then DAQ score is available in the GitHub. Then in this GitHub page, there's a link to access the Google Cloud version of the DAQ score. So one thing I should note that the Google Cloud have a 4 hours limit for free users. So if you pay some $9 per month, so you can use more. But if free user, you can use the 4 hours limit. Then this Google Cloud have only 2 CPU cores and slow GPUs. So if you want to try the bigger one on the bigger structures, please go to the website server, EM server. Okay, I'll show briefly the instruction. So in the GitHub page, so there's a link, 2 links. So online server is this one, then Collabo website is here. So you can simply click this link. Then there's a button open in Collab. Then in the Collab, Google Collab, so there's a runtime. It's a tab, runtime, then run all. So basically that's it. They automatically install all deep learning package, PyTorch, then Then some step, it stops the process. So please upload your PDB structure, atomic model here. Then click here, you can upload the PDB structure. Then later, this Google Cloud asks, please upload your EM map. Then click here, you can select the EM map. Then check the structure. That is relatively easy. So I will show you how to do that. Okay, can you see my screen? Is it okay? It's too small. Okay, so yeah, first you can search the DAQ score GitHub like this. DAQ score GitHub. Then maybe at the top, so DAQ GitHub link should be there. Then click maybe bigger character, okay. Then click here. You can see that online server. Yeah, later I will show. Then collab website. Then click this link. Okay, network. Oh, it's slow. Okay. Then here, yeah, you can see the open in collab. Then this is the Google Cloud version of the DAQ score. So then runtime. Here's the runtime tab. Then run all. If you ask something, then run anyway. Then now you can see that connecting the GPUs. Then it's assign the GPUs. Then it's running this process. So here is a green dot. So if you click the green dot, then it can automatically jump to the current process. So now the Google Cloud installing the packages and the programs. Yeah, it's installing the lot of packages. Okay, it will take 1 or 2 minutes. So far, do you have any questions? Yeah, any questions, fine. Technical questions or algorithm questions? Okay, so then now this Cloud page asking the please upload your PDB file. Oh, okay. So choose files. Yeah, basically you can upload any PDB structures. So in this case, I will upload the 7fet.pdb file. Then opens. Then it's uploading the file. Then next, input your EM map. Then choose file. In this case, 31563. This map is taken from the EMDB, then opened. So because this map is a relatively huge map, so it will take 20 or 30 minutes. So I will show the results page. So how it looks like. Yeah, I already computed this morning. Then computing automatically. Then at the end, it shows the 3D structure. So this target is a homotrimer, so 3 chains. So it's completely symmetrical structures. But the interesting thing is that the one positions, can you see that this red beta structure. Oh, okay. DAQ score say these positions is highly possible that's the wrong modelings. So it's something wrong. Then, yeah, it showed the DAQ scores for each chains, for A chain, B chain, C chains. So interestingly, the B chain around the residual number 600, it's a significantly low DAQ score. It's a minus negative 1.0. Yes. So like this, you can upload your EM map and PDB structures. Then you can evaluate the PDB structures. Okay, let's do some additional things. Okay. After computing the DAQ scores, so Google archive the all results, then provide the result as this one folder, input eval. So in these directories, you can check the individual chain structures and how it looks like by your cameras or PyMo. On the PyMo, so you can type this command, spectrum B, red, white, blue, O, -1 to 1s. Then you can check the very details on your laptop or PyMo sessions. Okay. Then I will demonstrate how to check the DAQ score on PyMo. Okay, maybe still, this process is computing. So PyMo, this is a PyMo, then file open. Then examples. So after computing the DAQ score, it will automatically generates these zip files. So in the zip files, there should be the 3 files, the DAQ score result. Then open the files. Yeah. Then type the same command, spectrum B, red, white, blue, O, -1 to 1. Okay. Is it difficult to see? Okay. Background white. Okay. Yeah, this is the result of the DAQ scoring. So we can see that these positions have very low DAQ score. So good thing is that in PyMo, so we can superimpose all 3 chains. So for example, the align to DAQ W9A. So by using this command, so we can superimpose all 3 chains to the same chain A. Yeah, overall structure looks almost identical, but somehow the chain B at these positions have a low DAQ score. That's much better. Yeah, actually, residue number 600 and 605. Then show the sticks. So yeah, this is very interesting. So both the chain A and C use almost identical structure. But at this position, only chain B have a different sequence assignment. So residue number 600 is modeled here. Then 605 are modeled here. Yeah, its sequence shifting happens. Even this PDB entry should be the homotrimer. So all three chains should have the same structures, but somehow author build a different structure at that positions. Then DAQ score say the chain B is very bad. So maybe this is correct. So somehow the author mismodeled that positions. So yeah, by using the PyMOS, you can confirm that the actual sidechain orientations and assignments, then evaluate by yourself. Okay, so then, so DAQ score is also available in the EM server. EM server. So EM server is addressed here. So HTTP EM Kihara.org. So it's faster and also no limitations. Yeah, please try to use this website. Okay, I've showed how to use that. Okay. So this is the Kihara EM web server. Kihara EM. So on the Google, so typing the Kihara EM maybe server, then Kihara Cryo-EM suite. Then server is available, EM2 server is available. Yeah, this is a link or EM2 server. Then click here. Yeah, different program is available on the server platform. Then DAQ score. The process is all identical, so you can select the protein structure like I did. So PDB structure, then choose the file, density map, then upload. Yeah, uploading the file. Yeah, it's easy. Then I will show the examples output files. So it shows the same result. This has a interactive plot here, so we can easily see that the chain B have something wrong. So this is a web server, EM web servers. So DAQ score is available on the Google Cloud and web servers. So this is much faster than Google Cloud, but if you cannot upload your new EM map, then please use the Google Cloud. It's confidential. But if you want to know the quick results, yeah, please use the EM web server. It's faster and easier. Okay. The next thing is the DAQ score database. So DAQ score database is available at this website. Okay, let's demonstrate the DAQ score database. DAQ score database. Yes, this is a DAQ score database. Click here. Then you can use any keyword, any EMDB ID or PDB IDs. Okay, let's search some structures. So do you have any suggestion to use keyword?
as a keyword? Do you want to search something? Anything's fine. Okay, then, okay. Apophylating. Okay. Okay, apophylating. Yeah, a lot of apophylating structures are available. Yeah, apophylating is almost all entries have our same structures. Yeah, but maybe we cannot find any significant errors. Yeah, for example, here, so these regions have a negative 0.5 values, but I think this is not indicating that's the modeling error. Maybe that positions have a very high local resolutions, then network confused that's the accuracy predictions. So 0.5 is a gray zone. Yes, but core regions have a very high qualities. Yeah. Apophylatings. Okay, nice, interesting example. Thank you. Okay. COVID-19, maybe COVID-19 is available. Yeah, we can search a lot of structure. If you click the entries, you can check the individual chains carefully and also the positions. It's interactive. Okay, so again, yeah, it should be noted that the DAQ score 0 means the density map itself does not have enough information. So maybe, yeah, here, this outside loop regions have a 0 DAQ score. So that means that maybe densities are not clear. So that's why the DAQ score is 0. But if the plot shows DAQ score shows negative 1.0, it's 99% long modeling. Yeah, please try to use this DAQ score database. Okay. Okay, so far, do you have any questions or something, comments? Okay, yes.
This only for protein chains or also
Okay, so currently it's only protein chain. Yeah, but we are developing the nucleotide or ligand. So later, yeah, I hope we can show some results. But currently protein structure only.
and when I upload the structure go back to remove the ligand?
Yeah, automatically remove the ligand.
Oh,
yeah. You don't have to worry about ligand or some membrane backbone. Yeah. Okay. Okay, so the next thing that DeepMainmast Google Cloud. So we implement the DeepMainmast architecture on the Google Cloud. So good thing is that DeepMainmast Google Cloud is free and confidential. So, but there's a 4-hour limitations, the 2 CPU cores, and also it has only 15 gigabyte GPUs. So if you want to use the bigger map, bigger structures, maybe the server or command line version is a better way. But anyways, yeah, I will show the Google Colab versions. So it's basically almost same with the DAQ score. Okay. In the page, the user can specify the job name. Then, if you don't want to use the examples, yeah, please do not check this checkbox. So default is not checked. So job names, then run the command, run the script, then ask the please upload your density map, sequence file. Density map and sequence file. Sequence file should be like this format. So chain A, this, chain B, this, chain C, this. Then if you already have AlphaFold2 models, so please check these buttons, then this script automatically asks please upload your AlphaFold2 models. Then parameters. So control level. So if you know some optimal control levels, then please specify. It will improve the computational speed. Otherwise, it will check all of the positions to view the structures. And number of seconds. So it is controlling the modeling accuracy. If you spend more time to compute the DeepMainmast, you can get better results. But if you want to get the quick result, you can reduce the number of the this second numbers. So in our server, we set the number as the length of the residues. So if you compute the 1,000 residue long, yeah, please set 1,000. But if you use only 50 residue longs, maybe 50 is enough. And also that your target has a homo oligomer part. Some part is a homo oligomer. Please check this. They try to define the chain assignment automatically. So this is the examples result of the 3D viewers. So because the Google Colab version have limitations, it does not build the entire side chains, full atom structures. So it generates only the C alpha positions. So after downloading the file, so you can check maybe A chain, B chain, C chain. Then each position have a DAQ score. So how confident of that modeling? So blue region is a very confident. The red region is a not confident region. Okay, let's do the demo. Okay, yeah, first, please access to the DeepMainmast GitHub page. Maybe, okay. DeepMainmast GitHub. Then it will show the Kihara GitHub page, then DeepMainmast. Yeah. Then there's a link for the EM server or Google Colab versions. So click here. It's almost same with the DAQ score cloud page. Okay, so yeah, it will take at least 1 hour or 2 hours. So I will show the result here. After computing the structures, it will show like this. Then you can download the structures. So basically, yeah, step by step, please click the buttons. Then you can upload a sequence, structure, alpha code structures, and EM map. Then after 2 hours, you can get this kind of structure. Okay. Okay, then next thing is the EM server, so DeepMainmast in the EM server. So we are also releasing the server version. So good thing is that it's much faster than Google Cloud. So we can use 24 GB memory GPU card and 8-core CPUs. And also it can build a full atom structure buildings, and also do the Rosetta local refinement. Then there is no CPU time limitations. So yeah, you can access to the website here. Then there are some examples I will show. The parameter is almost same. It's very hard to read. So, but it puts the control levels and Cryo-EM map, then faster files. Then if AlphaFold2 structure is available, you can simply put this here to the AlphaFold. If you want to build the Rosetta full atom building and refinement, please check these buttons. It's very straightforward. Okay, I will show the here. So this is the GitHub EM server. Then click here. The server input example input website. Then select the EM map. Uh, 2513 opened. Then first file opened. Then AlphaFold structures. So this case, I prepared AlphaFold structures. So if you have 3 chains, you can just simply concatenate the all AlphaFold structures. You don't have to worry about the chain ID or region numbers. You just concatenate the 3 models as a single PDB file. Then program automatically identify that, oh, this is the A chain, this is the B chain, this is C chains, or local positions, like so on. Opened. Then this case, it's a heterotrimer. So I want to compute the full atom structures, then upload. It's running. Yeah, it's running. Yeah, some jobs are running. Maybe I have an example of that. Okay, this is the example of the output files. So it took 2 or 3 hours on our websites. But we can see that the side chains also available. It's already optimized, no crash, and optimized on the density map. It's relatively easy. Yeah, if you have a density map and sequence, please try to use this DeepMainmast web sub. Okay, it's almost done. Then, final things is this ones. So last week, we also released the command line versions. So because some users have a very high-end GPU cards and also a lot of cores. In that case, by using the command lines, so you can specify the number of the CPU cores and threads, then you can get the structures much faster. So there's no RAM limitations because your machines have RAM. Then you can use the local GPUs and local CPUs. So very detailed instructions are available this GitHub page. So today, I cannot show the very detailed things. So but yeah, please access to this GitHub page. And the good thing is that in Hochmeyer buildings, this DeepMainmast is already installed. So if you want to use your local machine, yeah, please try the module load conda 38, then do this command, then module load DeepMainmast merge. Then download some files, git pull, git clone command. Then you can run the latest versions of the DeepMainmast on your machines.